Now, moving on to the second specification of this unit. Specification number 2 requires us to identify and label the different parts of a dicotyledonous leaf as seen in diagrams or under the microscope, what we call photomicrographs. But it doesn't stop there. We also need to explain how each of these structures is specially adapted to carry out photosynthesis and gas exchange effectively. There are 6 key components we need to focus on. First, the stomata and guard cells, which are responsible for controlling gas exchange and water loss. Then we have the spongy and palisade mesophyll cells, which play different roles in maximizing photosynthesis. The air spaces between cells allow for efficient diffusion of gases. We'll also look at the vascular bundles, that's the xylem and phloem, which transport water, minerals, and food throughout the plant. Another important feature is the distribution of chloroplasts, especially in the palisade layer where light absorption is maximized. Finally, we have the upper and lower epidermis, which help protect the leaf and regulate water loss. To help you understand all this more clearly, we'll be looking at diagrams that show a cross-section of a leaf. You'll be able to see each of these layers and structures clearly labeled. And not just that, we'll also use real photomicrographs so you can see what these structures actually look like under a microscope. So, let's begin exploring the inner structure of a leaf and see how it's beautifully designed for its role in photosynthesis and gas exchange. When we talk about leaf structure, it's important to understand that leaves can be categorized based on their internal anatomy. Broadly, there are 2 main types, monocotyledonous leaves and dicotyledonous leaves. Now, if you take a look at the diagrams presented, you'll notice they show cross-sections of each type, highlighting their internal structures for comparison. Starting with the monocotyledonous leaf, the diagram on the left, you'll see that the vascular bundles are scattered throughout the leaf, and the mesophyll tissue appears fairly uniform, without a clear separation into different layers. In contrast, when we examine the dicotyledonous leaf, things look more structured. The diagram clearly shows a distinct upper and lower side, with palisade and spongy mesophyll layers differentiated. The vascular bundles, including xylem and phloem, are arranged in a well-defined pattern. Since our syllabus focuses more on the structure and function of dicotyledonous leaves, we'll narrow our attention to this type. So from here on, we'll be exploring the internal anatomy of a dicot leaf in detail. Alright, now let's take a closer look at this detailed 3D cutaway diagram of a dicotyledonous leaf. It's a really useful visual that ties directly into what we need to learn for our syllabus, highlighting all the key structures and where they're positioned. Right at the top, you'll notice the cuticle, that's the thin, waxy outer layer. Its main job is to reduce water loss from the surface of the leaf, especially under hot or dry conditions. Beneath that, we have the upper epidermis, which is the protective layer of cells on the top surface. Similarly, at the very bottom of the leaf, there's the lower epidermis, which serves the same protective function. Between these 2 outer layers lies the mesophyll tissue, and this is where most of the photosynthesis action takes place. The mesophyll is divided into 2 parts. First, there's the palisade mesophyll, just below the upper epidermis. These cells are long, tightly packed, and packed full of chloroplasts, which are the little green structures you can see in the diagram. The job, capturing sunlight for photosynthesis. Below that, we have the spongy mesophyll. These cells are more loosely arranged and have large air spaces between them. And that's not by accident, it's a brilliant adaptation for gas exchange, allowing carbon dioxide and oxygen to diffuse easily in and out of cells. Now, if you look at the lower epidermis, you'll see tiny pores labeled as stomata. These are the entry and exit points for gases. Each stoma is flanked by guard cells, which open and close the pore to regulate gas exchange and water loss. Also labeled in the diagram are the vascular bundles, which include the xylem and phloem. These are the plant's transport systems. Xylem carries water from the roots up to the leaf, while phloem transports sugars made during photosynthesis to other parts of the plant. And one more important point, the chloroplasts aren't randomly scattered. The diagram shows they're mostly concentrated in the palisade mesophyll, where they get the most light. But you'll also find some in the spongy mesophyll and even in the guard cells, all contributing to photosynthesis. Finally, this is how these structures help the leaf to do its jobs such as photosynthesis, water transport, and efficient gas exchange. It even controls gases like carbon dioxide and oxygen move in and out of the air spaces and the surrounding cells. So, this diagram isn't just a picture, it's a map to understanding how a leaf works like a tiny solar-powered factory.